Hello, everyone. Welcome to today's live broadcast, Protect Your Cells with Proper Pipetting, How Liquid Handling Influences Your Cell Culture Work. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Eppendorf. Eppendorf is a leading life science company that develops and sells instruments, consumables, and services for liquid sample and cell handling in laboratories worldwide. For more information, please visit eppendorf.com. We have a few important announcements before we begin. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. This webcast is also designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window, or submit your problem through the green Q&A button lower left. I would now like to introduce today's speakers, Jessica Wagener, PhD, and Hanae Henke, PhD, both from Eppendorf's headquarters in Hamburg, Germany. Dr. Wagener is a field application specialist focusing on cell handling. Entering a cell culture laboratory as a graduate student for the first time in 2006, she came to Eppendorf with 5+ years of experience in cell culture. After studying biology at the universities Marburg and Dusseldorf, Jessica investigated apoptosis mechanisms in different cell lines for her diploma thesis. Following 1 year as a research associate working with primary cells, she started to work toward a PhD with a focus on reproductive biology at the medical school Essen. Jessica joined Eppendorf in 2012, shortly after completing a PhD in biology. Dr. Henke is a field application specialist focusing on liquid handling, specialized in positive displacement dispenser and pipette. She joined Eppendorf in 2015 after finishing a PhD in medical microbiology on the topic of Staphylococcus epidermis hip joint infections at the University Hospital in Hamburg. In vitro macrophage studies were part of her PhD research. Hanae focused on molecular biology and genetics early during her studies of biology at the University of Hamburg and deepened her knowledge in food microbiology laboratories and at Harvard Medical School in the group of Professor Roberto Kolter. I will now turn it over to Dr. Henke for her presentation.
Thank you very much for the kind introduction, Judy. Good morning, good afternoon, and good evening, everyone. Thank you for joining us to this webinar, Protect Your Cells with Proper Pipetting. We will discuss how liquid handling influences your cell culture work. As Judy said, my name is Hanae Henke, and I will start going through the fundamentals of pipetting before my colleague Jessica Wagener will go through all the cell culture relevant topics. The webinar covers 4 main topics. First, there will be an introduction into pipetting fundamentals. Then contamination prevention will be discussed before we get into detail on pipetting in cell culture. In the end, you will get information from cell seeding to cell banking and a summary. Everybody working in a lab is pipetting every single day. And as most things we do every day, we do not think about it much. But when it comes to this critical step in an experiment, everything can be changed, and minimal differences in pipetting can decide on a result being precise and satisfying or disappointing. Furthermore, accurate pipetting technique is necessary when publishing data. So everyone following proper pipetting rules can redo the experiment and verify your results. Actually, as we are pipetting so much, it should be one of our major concerns, and then it shows having the highest impact of all techniques on our experiments. On the start of each experiment stands the choice of the correct pipetting tool. So let us have a look on the different devices one can use. The different pipette forms vary in their handling, precision, and throughput. The precision ranges from manual over electronic up to automated systems. What is the reason for that? So, in manual pipettes, the technique and the speed is under user control, while in electronic pipettes, The technique is under user control and the speed is under device control. So for automated systems, both technique and speed are under device control and most precise. Each pipette tool is ideal for one application. For example, a pipette helper with serological pipettes for large volumes, like the transfer of liquid from cell culture flasks, medium bottles, or conical tubes during the exchange of culture medium. Single and multi-channel pipettes are ideal for applications where the precision is more important, like thawing of stock cultures or subculturing. When it comes to small volumes, less than 1 milliliter, the requested precision and accuracy are much higher, and these small volumes are mainly used in cell seeding, addition of compound and assay reagents, as well as when working with cryovials and microtubes. When using multi-well plates, a dispenser or multi-channel pipette makes sense to add medium and compounds fast and precise. Dispensers can be used from 1 microliter up to 50 milliliter by using the same device and just changing the tip size. So what we see here is that each device has its optimal tip. They all look different and they are all optimized for different applications. Additionally, it is recommended to use the tips manufactured by the same company as the pipette tool, since only then a perfectly working system can be guaranteed according to the ISO 8655. One special challenge in cell culture is pipetting of problematic liquids like cell culture medium that builds up a lot of foam due to the high protein content. Viscous liquids like DMSO are not only used as cryoprotectants, but also in a lot of cases as solvents, for example, for drugs or inhibitors. Due to its viscosity, a large amount of liquid can remain in the tip when pipetting DMSO, which leads to false concentrations and a lot of waste of this chemical. Also, ethanol is quite often used in cell culture labs, either as disinfectant or a solvent for any lyophilized components. But its high vapor pressure makes it difficult to pipette correct volumes, since the ethanol tends to drip out of the pipette tip after aspirating. To deal with these liquids, we will take a look on different pipetting mechanisms. But first of all, we would like to ask you a question. And we would love you to answer it. So the first poll question will be, which pipetting instruments do you use for cell culture? You can choose between manual and or electronic dispenser, manual and or electronic air cushion pipettes, electronic pipette controller, or classic pipette controller, like a Pileos ball. So we will wait a few more seconds until everybody answered the question. I hope that most of you made their choice already. Okay, so we will close the question. And we will see the results in a few seconds. Oh, wow! So it's definitely that most of you are using manual and or electronic dispenser for cell culture. 68.5% already. This is very good. About 21% are using manual and or electronic air cushion pipettes. Only very few are using electronic pipette controller, and some are still using the classic pipette controller. So we will have a closer look on the different mechanisms now. So the classic pipettes are air cushion pipettes. The name derives from the air inside the pipette that is between the actual sample liquid and the instrument's piston, as you see on the picture here. When the air is pushed out by pressing the top button, according to the amount of air, the liquid is soaked into the pipette tip when the top button is slowly released inside the liquid. As you can see, the liquid has direct contact with the air inside the pipette, and therefore the formation of aerosols can lead to contaminations inside the pipette. We will talk about aerosol formation in a few minutes. This system is widely distributed and only suitable for aqueous solutions since these pipettes are calibrated to water. Problematic liquids with a high density, like DMSO or high vapor pressure, like ethanol, cannot be pipette accurate without a special technique like reverse pipetting or a readjustment of the pipette. Less sensible are positive displacement pipettes, also known as stepper or dispenser. Here the liquid is soaked into a tip with an integrated piston. There is direct contact between the liquid and the piston. There is no air cushion. So the risk of contaminating the pipette from the inside is eliminated. This system allows the transfer of all liquids no matter if they have a high density or high vapor pressure. Dispensers are more precise and more accurate than air cushion pipettes with problematic liquids. The tip of the dispenser is filled once and then the liquid is dispensed in multiple steps. It is especially suitable for cell culture when working with plates, so to work faster and more precise than with air cushion pipettes. So all of you who are already using the dispensers, that's very, very good. If you want to know more about pipettes and pipette calibration, Eppendorf also offers liquid handling seminars in many different countries, or you may watch the webinar on pipette calibration that is coming up on September 20th. So now I would like to hand over to Jessica for the contamination prevention best practice.
Thank you, Hanae. Hello also from my side. In this next chapter, I would like to talk about some general aspects of reducing the risk of contaminating your cells. But before I start, here comes our next poll question, and we ask you to participate by clicking. Did you ever have problems with contamination in your cell culture labs? And the answers can be never, rarely, sometimes, or on a regular basis. So again, we will wait some seconds until the answers are coming in, and then we will push the results. Okay, so the results are coming in right now. So as you see, looking at these poll results, we can see that only about 3%, only about 8% of you never see contamination of their cells, whereas the majority, around 50%, and also 41% of you are facing contamination sometimes or even on a regular basis from time to time. All right. So contamination is maybe the biggest challenge when working in a cell culture lab. In general, we find two major types of contaminants, the microbial ones and the eukaryotic. The microbial contaminations are fungi and yeast, bacteria, and here especially mycoplasma. The other ones, the eukaryotic, are not necessarily so well known and are cross-contamination of other eukaryotic cells in cell lines. This phenomenon was first described in 1967, and to date it is known that about 15 to 30% of cell lines used in laboratories worldwide are either cross-contaminated or misidentified. No matter what type of contamination we are talking about, I think we can all agree on the consequences. The loss of time, since you have to repeat experiments, the increase in cost, time is money and more material is needed. The loss of reproducibility, you might have completely different responses when you work with contaminated or non-contaminated cells. And the last point mentioned here might be the most horrific, the loss of significance. Just imagine if you want to find a new therapeutic treatment for breast cancer, but you work with melanoma cells without knowing it, the results of your work are unreliable. So taken together, avoiding contamination is much easier than dealing with it. There are so many possibilities of getting contamination, so prevention is always the logical step. Maintaining a sterile environment has first priority in a cell culture lab. Instead of sterile, I should better call it germ-reduced environment, because this is the actual truth. For example, the inside of an incubator is never sterile. Every time the door is open, germs can enter the incubator, plus the atmosphere inside the incubator is disturbed. Therefore, door openings should be reduced in duration and frequency as much as possible. Preventive hygiene plays a big role in preventing contamination. That means regular cleaning of lab equipment like incubators, water baths, biosafety cabinets is a must. Also, personal protective equipment is very important. You should have a dedicated lab coat that you only wear inside the cell culture lab. Every other type of personal protective equipment, like for example lab shoes, further reduce the risk of contamination. Your working place in the biosafety cabinet is the place of action, and the place where most mistakes can happen. I will come back to that point in more detail later. All preventive hygiene and personal protective equipment and organization is useless when you don't take care of your aseptic technique while handling the cells, reagents, and last but not least, pipettes. The person who is working in the cell culture lab is maybe the biggest source of contamination. Your hands are the major source of germ transfer when handling the cells. It might be trivial to specifically mention, but when entering the cell culture lab, the first thing to do should be washing your hands. Disinfecting your hands before putting on gloves further reduces the contamination risk. Another advice is to use hand cream regularly, because healthy skin has fewer germs than dry skin. When you put on gloves, disinfect them sufficiently. That means let them dry before you start working. And disinfect them not only once, but repeatedly. For example, when you adjust your glasses, never do it without subsequent hand disinfection. You can also tuck the sleeves of your lab coat into the gloves to further reduce the risk of contamination. I already mentioned the importance of having a lab coat that is reserved for the cell culture lab. Same applies, of course, for any other type of equipment. Especially pipettes and tip boxes are easy to carry from one lab to the other, which should be strictly forbidden. A good tip is to simply put a sticker on each pipette and label it with cell culture, so everyone knows that this pipette should be kept inside the cell culture lab. Disinfect the pipette each time before you place them in the biosafety cabinet. Do it thoroughly by wiping with a disinfectant to allow a complete wetting of the surface. If you choose a new pipette for cell culture, consider one that is easy to clean with a smooth surface that does not have any rough edges and grooves. You can also disassemble the pipette to clean them from the inside if necessary. Autoclavable pipettes give extra safety. When you are pipetting, try to avoid moving your hands or arms over open vessels. At the beginning of this chapter, I mentioned the importance of having an organized working place in the biosafety cabinet. As this is the area where you open the dishes, plates, or flasks with cells, there are some things to be kept in mind to avoid contamination. You should always work in the middle third of the working space, not too much in the front, not too much in the back, because there you disturb the laminar airflow in the cabinet. Another advice is to not block the air grills in the front, the back, or at the sides, with research notes, discarded plastic wrappers, pipetting devices, and so on. Disinfect the working area in the hood completely and let it dry before every use. Disinfect every piece of equipment that you bring in. Every item that is in there can disturb the laminar airflow that keeps the germs outside. So don't overcrowd your working space with items that you don't need for that particular working step. Any droplets or spills need to be cleaned immediately. When working at the hood, it is a good advice to have one clean side with culture media, pipettes, and new consumables. The other side is reserved for liquid waste, used tips, and so on. And with this, I will hand over to Hanae again, who will proceed with the next chapter, pipetting techniques in cell culture.
Thank you, Jessica. And now let us have a look on what is most important in cell culture experiments. The first thing is a protection. In the following slides, we use protection as a term to describe that, of course, all steps have to be performed under sterile conditions, but some methods are safer than others. For example, the distance to cells or reagents, like a serological pipette with a 30 centimeter distance to potentially non-sterile hands and electronic pipetting controller dispensers with only 10 centimeter distance, or even a pipette with a tip that only has 4 centimeter distance. So if you compare these three things, a serological pipette is usually safer than the others, even when special precautions are taken. The second term is precision. This is the term we use in all slides, but we actually refer to the validation terms precision and accuracy with this word. So an explanation would go beyond the scope of this presentation and is part of the pipette clinics. And the third topic we will talk about are the times, the time and cost. So the pipetting of a 96 well plate with a single channel versus a multi-channel pipette is a good comparison of speed and therefore also the cost per sample. Now I would like to start with the main topic of protection, aerosols. What are aerosols actually? Aerosols are formed always when a liquid is transferred, especially when using warm liquids like cell culture medium at 37 degrees. In room temperature conditions, a slight evaporation leads to very small aerosol drops that can contaminate the inside of the pipette and with this lead to cross-contamination of the samples. As you see in the illustrated pipette tip, an effective barrier against aerosol formation is a filter tip, compared to not using a filter tip. It stops small aerosols and contaminants such as bacteria or virus to get into the pipette. However, using a dispenser with a fresh tip every time is 100% contamination-free and the more valuable choice when it comes to sensitive samples. Aerosols do not have a chance in dispenser tips. Let's have a closer look on filter tips. When using filter tips, remember to use a two-layered filter. Its first layer retains drops, splashes, and large particles like bacteria. The second layer retains small aerosols, biomolecules like DNA and RNA, as well as virus particles that could contaminate the pipette cone and cross-contaminate your samples. But not all aerosols can be retained by a two-layered filter tip. There is still a chance for very small aerosols to successfully pass the filter. Therefore, using a positive displacement system is more secure. Furthermore, some tricks to avoid aerosol formation are mixing by gentle rolling rather than vigorous shaking of a reaction tube. Pipetting directly into a liquid or onto a surface avoids bubbling and splashing of liquid, and placing the containers, in this case I mean reaction tubes, very close to each other when transferring the liquids between them avoids drops that fall or splashes from one tube to the next. And remember to slowly remove tube caps or stoppers. So let us go over to the next big topic, the precision. What did we learn about precision in pipetting so far? One widely used tool is an electronic pipette helper. But keep in mind that the liquid volume is mainly defined by the person using the electronic pipette helper. The meniscus is seen by the person pipetting and stopped when the volume on the graduation is reached. This is a very personal system, but least precise. A more precise option is a manual pipette. These are suitable for aqueous solutions, but still the technique and speed fall to the user. With electronic pipettes, the speed device is device controlled and only the technique is the precision limiting user-dependent factor. The highest precision with all liquids is achieved by using dispenser. The device dispenses the same amount of liquid on each stroke, either manually or electronic. So let's have a closer look on each tool and see for which application and which part of cell culture it is mainly useful. For large volumes where precision is not so important, an electronic pipette helper is sufficient. For example, when exchanging culture medium. When using flasks, it might also be of advantage to use serological pipettes, elongated tips, or a dispenser that comes with long tips to avoid contact of the pipette with the inside of the tube or flask. You see on this picture that the pipette cone reaches directly into the tube, which leads to contamination. So imagine you have a cell culture and you always have these little drops up on the tube, on the conical tube in this case. You will touch it with your pipette and then just carry it over to the next tube. So another important thing is choosing the right vessel according to the application. The pipette tool used is as important as the whole experimental setup. If you're using a manual pipette, the tube used should not be too high that you touch the inside of the tube. So the 5 milliliter tube is safer to be used with a manual pipette instead of the 15 milliliter tube or a flask. And using a 15 milliliter tube with a dispenser and the according tip is a safe option. You do not get the device in contact with the inner side of the vessel and protect your sample as well as the device. As you see on the picture that just popped up, only the dispenser tip reaches into the 15 milliliter tube, and as you know, the tip will be discarded after usage, so you always have a fresh, sterile tip for your experiments. When pipetting upright over open flasks for the aspiration of cell culture medium or used cell culture medium, contaminations may fall into the open flask as you see on the right side. So remember to always use a 45-degree angle when pipetting out or into vessels with a pipette controller. When it comes to small volumes, multiple things have to be considered. The precision is the most important of these things for all the assays or the addition of compounds, because even a little difference in the volume you pipette may lead to cell death or another reaction in your cell culture. Therefore, a manual or electronic pipette or a dispenser are most suitable. I would recommend a dispenser in this case to be absolutely sure. But if you're using a manual air cushion pipette, there are some other tricks you should consider. On these illustrations, you can see two major things to remember during aspiration of liquid. First of all, the immersion depth, meaning how deep you put the pipette tip into the liquid. And second, the angle you are using when holding the pipette. Diving in too deep raises the surplus liquid volume inside the pipette tip, so you aspirate too much liquid. Additionally, holding the pipette at an angle of 30 to 40 degrees raises the surplus liquid volume even more, and you result in an imprecise pipetting result. Ideally, holding the pipette upright vertical and an immersion depth as shallow as possible leads to the most accurate liquid aspiration possible. So keep in mind that for liquid volumes below 20 microliter, a very low immersion depth is recommended, so only as much as necessary. While for liquid volumes above 20 microliters, you might immerse the tip a little deeper, but never more than 5 millimeters. So now I would like you to answer another poll question. So the third question will be, do you use a dispenser for small volumes already? And you have three different answers: yes, no, or I'm not sure how to work with these devices. And as many of you already answered that you're using a dispenser, I think the third answer will not be covered that much. So I'm curious on how many people are using the dispenser for small volumes actually. We're waiting just a few seconds until everybody answered. Okay, so most of you answered, and Now we want to have a look on the results. So, let me see. All right, so most of you, over half of the audience, is not using a dispenser for small volumes. Around 30% are using a small dispenser for small volumes already, and still 13% are not sure how to work with these devices. So if you want to know a bit more about dispensers, I can only recommend you the webinar Solutions Beyond Water. There, it's also from Eppendorf, and we describe the method of using a dispenser a bit more in detail. But now I would like to go over to small volume dispensing and some more tips and tricks on doing it right. So when dispensing the liquid at a 45-degree angle and securing the filled tip on the wall of the vessel, it's absolutely fine. If you're using an air cushion pipette, it's actually mandatory. A blowout is also absolutely necessary when using manual air cushion pipettes. So with dispensers, a blowout is not necessary since each dispensing step delivers the correct liquid volume. If you maybe have concerns using a dispenser for small volumes because you have to aspirate more liquid than you actually want to dispense, I can only recommend to use an electronic dispenser. There you have a pipette mode where you only aspirate and dispense the amount of liquid you really want to use for your assay, so you do not have waste of reagents. While dispensing, avoid an upright position to not stress your cells additionally by liquid directly spotted on the cells. Do not move your hand over the plate to avoid contamination. So now let's have a look on the tips for small volumes, especially with a dispenser. So using dispensers make the whole pipetting process easier, and you do not have to think about immersion depth or the aspirating angle. Additionally, you can use any liquid with a dispenser, no matter what property the liquid has, like viscosity, density, or vapor pressure. You could also dispense in free jet, which is not recommended with manual air cushion pipettes. And as I just mentioned, use the pipette mode in an electronic dispenser to avoid having residues of your reagent. So now I would hand over to Jessica again for the last part and the summary.
Thank you, Hanae, again. So in this last chapter, I want to give you a few practical tips on some of the most critical steps in your cell-based experiments. Let's have a closer look at this first experimental step that is so important, the seeding of cells. The precondition for getting comparable results is to seed out the same number of cells for every negative control, positive control, and for every experimental replicate. The more replicates you include, the better for your statistical analysis, of course. Variations in seeding cell numbers and the formation of air bubbles during seeding will not only increase standard deviations, it will also make your results less reliable. Repeated mixing of the cell suspension during seeding and careful pipetting can be a solution. Let's get more into detail on the next slide. When you fill, for instance, a multi-well plate with cells, it will take some time to fill all wells. You can use a single-channel pipette and have the cell suspension in a tube, or you can use a multi-channel pipette and have the cell suspension in some type of reservoir. No matter which tool you choose, the longer the seeding process takes, the higher is the variation in cell number from one well to the other. As you see in this little cartoon here on the right, the cells sink towards the bottom of the tube with time, and the sedimentation goes quite fast, within minutes. Therefore, you need to resuspend or mix the cells to swell them up in between and get an equal number of cells in every well. Saying that, we should keep in mind that cell culture media tend to foam due to the high protein content of supplemented serum. Air bubbles can hinder cell attachment, so you should avoid harsh and fast pipetting to reduce the formation of air bubbles and also to reduce shear forces, which further stresses the cells. Now let's have a look how pipetting can affect your assay results. In this graph on the left, you see the results of a cell viability assay. Each column represents one row of a 96-well plate. The aim was to seed an identical number of cells in each wells. 2 rows were left out for blanks here. The cells were seeded with a manual 8-channel pipette from a reservoir. After 24 hours, the number of viable cells was determined using a colorimetric assay. In rows 1 to 5, you see varying cell numbers in the wells and therefore high standard deviations. Here the user did not mix the cell suspension between pipetting each row, so the cells sedimented in the reservoir with time, and in addition, the pipette was held in different angles and heights during aspiration. In rows 6 to 10, you see the results with mixing the remaining cells before pipetting each row, aspirating at constant height and in upright position. Taken together, these data show quite impressively that the pipetting technique has a big influence on the results. You can also use a dispenser instead of a multi-channel pipette, of course, for applying assay reagents or seeding cells. With a dispenser, immersion depth and pipetting angle do not influence the precision. With this next slide, I want to talk about another very important step in cell culture, the cryopreservation of cells. When you want to establish frozen cell stocks of high quality, the stress factors need to be minimized. Cryoprotectants like DMSO are harmful to these cells in non-frozen media. When cells are freshly thawed, they are very sensitive to shear forces. So taken together, time is a critical factor when freezing and thawing cells. When you freeze the cells, you should be quick from the point where you add the DMSO-containing freezing medium to the cells until you start to cool them down. The cooling procedure itself should then be slow to avoid the formation of ice crystals. To achieve a cooling rate of 1 degree per minute, use a special freezing container and put the cells into a -80 freezer for 24 hours. You should not store the cells for longer time at -80, but freeze them in a liquid nitrogen tank. When you thaw the cells, time is again critical, because you want to minimize the exposure to DMSO. So thaw the cells as rapidly as possible and dilute them to decrease the concentration of DMSO in the medium. After that, you can either remove the DMSO directly by centrifugation or dilute the DMSO further and change the medium the next day. Saying that, it is important to mention that freshly thawed cells are much more sensitive to shear forces, so too harsh pipetting again should be avoided. In general, it is a good advice to not work with too many samples at a time and be organized. For example, label cryotubes or culture vessels first before you start to prepare the cells for freezing or thawing. I want to end this chapter with some general remarks about cell banking, because having high-quality cell stocks is your backup system and the prerequisite for reliable research. The most important point is that you make sure that you don't freeze contaminated cells and that the cell line is really the cell line you think it is. So authentication of cell lines should be performed regularly, especially when you got the cells from another lab and when you use them in long-term or subsequent studies. The cells you are working with should also be tested for mycoplasma regularly. As Hanae mentioned earlier in this presentation, filter tips give extra safety, so they should be considered at least for such important steps like cell bankings. The routine use of antibiotics is not recommended because they can mask a contamination with partially resistant bacteria. And please keep in mind that antibiotics are not effective against mycoplasma anyway. A two-stage cell banking system with a master and a working stock provides you constantly with early passages from your cell lines. A detailed record of the cell stocks is as important as clear labeling of cryotubes. So, to summarize what we were discussing in this webinar, here we have again the 3 aspects that are important when talking about pipetting in cell culture: precision, protection, and time. When a precise volume is needed, for example, when pipetting assay reagents, it is important to hold the pipette upright during aspiration and to use a constant immersion depth. When you empty the tip, rest it on the vessel wall and blow out the liquid completely. When sealing cells, for example, in a multi-well plate, it is very important to mix the cell suspension in between pipetting steps to achieve equal cell numbers and enhance the reproducibility of your experiments. But make sure that you avoid the formation of air bubbles. Pipettes are precision instruments. To keep the precision, it is essential to calibrate them on a regular basis. The second aspect we were discussing is protection against contamination. You can reduce the risk of contamination when you hold the pipette in a 45-degree angle, so that you don't have your hand directly above the open vessel. For example, when you are feeding or subcultivating the cells, protection is more important than a precise volume. Of course, all pipetting tools used in cell culture should be thoroughly disinfected before each use. It is also good to autoclave pipettes regularly, but please check with the manufacturer first if the pipette you are using can be autoclaved. Having a dedicated set of pipetting tools that don't leave the cell culture lab and using filter tips, at least for the very important steps like freezing cells, help further to protect the cells against contamination. When handling cells and media in the biosafety cabinet, the working space should be organized. Don't overcrowd the space with items that you don't need. Work only with one cell line at a time and separate clean pipetting tools, tips, vessels, and media from liquid waste, used tips, and so on. Last but not least, time can be a critical factor. For example, the time that it takes for filling all wells of a 96-well plate. Using multi-channel pipettes or a dispenser can save you a lot of time. Dispensers are a good choice for problematic liquids like DMSO or cell culture media that tends to foam. Remember how important it is to avoid the formation of air bubbles when sealing the cells. With a positive displacement system like a dispenser, you avoid air bubbles and you can be very quick in filling multiple wells or dishes. In general, you should not work with too many samples at a time to avoid that the cells are too long at room temperature, or to reduce the exposure to DMSO when freezing or thawing cells. Before we come to our last slide, here comes our last poll question. So please let us know how you rate the content of this webinar. Again, we will wait a few seconds until the answers are coming in. Thank you for attending this webinar. So thank you very much. So with this last slide, we want to point out the Eppendorf Training Center as a resource where you can find information about webinars and on-site training courses for different topics, including cell culture and liquid handling. So just go to eppendorf.com/etc to find an overview. This is the end of our presentation. Thank you for your attention so far, and we will need just a minute to go through your questions, and then we will start our question and answer session.
Thank you both for the informative presentations. Before we get started on the question and answer session, I'd like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can, and we'll follow up with any we did not get to today. Our first question is for Hanae. Can I autoclave dispenser tips for reuse?
So thank you for this interesting question. Actually, the components of the dispenser tips are of different materials, and we cannot guarantee the optimal function after autoclaving. So I have to say you cannot autoclave them.
Okay, the next question will be for Jessica. What is the best test method for mycoplasma?
It is hard to say really this is the best test methods for everyone, because there are a lot of different test methods out there. For instance, you can use DNA staining like DAPI, which is a very low cost method and a very quick method. On the other hand, the disadvantage here is that the interpretation of the results can be tricky. So there are also methods like PCR, for instance. There are some very reliable PCR kits out there So when you consider what type of methods you use, you have to consider how many samples do you have that you want to test for mycoplasma, how long can you wait for the results. For instance, you can do a microbiological culture really of the supernatant of the culture that you want to test. But then this is a very reliable method, but then you have to wait a few weeks until the results are there. So in that few weeks, the potential contamination has time to spread throughout the lab. On the other hand, you have to consider what type of equipment do you need. For PCR, of course, you need a thermocycler. There are also biochemical or ELISA-based assays out there, so you need the appropriate equipment for that. So there's advantages and disadvantages to everyone. I would recommend maybe a PCR because I think a thermocycler more or less belongs to the standard equipment of every lab now, and this is a very reliable method.
Okay. The next one is for Hanae. What is reverse pipetting?
Well, reverse pipetting is actually another pipetting technique than the most known forward pipetting. So this pipetting technique allows you to pipette problematic liquids, for example, with a higher viscosity than water, when using a classic air cushion pipette. Therefore, you push the operation button down to the second stop and aspirate the liquid, including the blowout volume. And when you dispense the liquid into your reaction vessel, you only press down to the first stop and do not perform the blowout. So some liquid will remain in the tip, but like this you get a more accurate pipetting result with viscous liquids than with the normal pipetting technique.
This one is for Jessica. How can I see the difference between bacterial and yeast contamination?
Bacteria and yeast look quite different when you compare them. So bacteria can have different shapes. They can be rod-shaped or spiral-shaped, or you can see little black dots. A lot of bacteria are actively moving, so this is not something that yeasts can do. Yeast are a little bit bigger than bacteria, still much smaller than the cells. They appear as round spheres, ovoid structures. They can be in chains or multi-branched, and they are usually have a bright color like white grayish, so they have a very characteristic appearance.
Thank you. If with a micropipette with a 0-degree angle aspiration is the proper way to do it, why is there a difference with large volumes aspiration?
Oh, thank you for this question. I already waited for this question actually. So the problem of aspirating upright or in a 45-degree angle is a consideration of precision or protection of the cells. So the most precise pipetting result will be achieved when the pipette is held perfectly vertical while aspirating. But your hand will be directly over the sample in this case. So you might risk a contamination. If the protection of the sample from contamination is your main focus, an aspirating angle of 45 degrees might be applied, but you will have a less precise pipetting in this case. So at a 45-degree angle, your hand won't be directly over the sample, and it gives you a higher degree of protection. But the negative effect, though, is a less precise pipetting result. So what I recommend for small volumes, most of the time the precision there is the most important point, and it already makes a difference if you aspirate 1 microliter of an assay component or only 0.8 microliters. But often with larger volumes, like when exchanging cell culture medium, it doesn't make a big difference if you aspirate 10 milliliter or 10.1 milliliter. So I recommend for large volumes where precision is not that important, you may choose the main part of protection and take a 45-degree angle, while for small volumes where the precision is essential, you should definitely stick to the vertical angle.
how long can we keep cells in -80°C during preservation?
That's a very good question. It of course depends on the cell type. So some cell types may be more sensitive to that temperature than others. In general, you should not store them at -80 for long term. So I would not do that for more than a few days actually. If it's more convenient for you to do that, I strongly recommend to definitely test the viability after you have them on -80 for a certain time, thaw them, do a viability assay, and compare that. Maybe do a time row, and then really take a very good look at the viability of the cells, how the cells behave. If you see an increase in cell death after you having them for longer time at -80. So you should avoid that to do it for long term or for more than a few days because it's not the appropriate temperature for storing cells. But it might be dependent on the cell type. So I would just test how the cells behave after you have them on -80 for a longer time.
What are the phenotypical observations for mycoplasma contamination in our cultured cell in plate or flask?
Well, you cannot see the mycoplasma. They are too small to be able to see them with a light microscope. So you might even not see any phenotypical or morphological difference in your cells because when you have the cells on a regular feeding schedule and you change the media regularly, which you should do, you are also feeding the mycoplasma regularly, and both can be in a balance. So it might be that you don't see anything. Still, the mycoplasma, although you don't see a phenotypical difference, still the mycoplasma have an effect on the cells. So they are growing mostly attached to the cell membrane. They can affect signaling pathways inside the cell. They can affect gene expression. So it's definitely recommended to test the cells regularly for mycoplasma, even if you don't see any change in the cells. So I would definitely recommend to test them regularly.
If sharing cells in foam formation is to be avoided, how do you get around this when you need to spin? Sorry, hang on 1 second. This question jumped away from me. Sorry. If sharing cells in foam formation is to be avoided, how do you get around this when you need to spin your cells down in a centrifuge and then resuspend?
That is also a very interesting question. I mean, of course, you have to centrifuge the cells in between if you want to have a definite cell number, and then you need to centrifuge, and then you also need to resuspend the cell pellet in medium. Avoiding the shear force and the foam is just by pipetting as gentle as possible, I would say, or use something like a serological pipette where you have a very wide opening so the shear stress is reduced. But in general, the best advice is just to pipette as gentle as possible, and when you seed the cells from the tube and you may have air bubbles on the liquid in the tube, then just try to not aspirate the air bubbles to transfer them in the dish or plate where you want to seed the cells. So try to leave the air bubbles in the tube if you cannot. You probably cannot avoid 100% of air bubble formation, but you should try to reduce it as much as possible.
What is needed for general maintenance and calibration of pipettes and pipetting devices?
Oh, the calibration of pipettes is a very sensitive, a big topic, and you would need, for sure, your pipettes, then the pipette tips from the same manufacturer. This is essential. You need a special room for the calibration with an analytical balance. You need distilled water, You need a special temperature between 15 to 30 degrees. The air humidity is not allowed to be more than 50%. So you need a lot of different devices like a hygrometer and everything to check all these requirements. And then the calibration of pipettes takes quite long because you have to measure the nominal volume, 50% and 10% of the nominal volume, each 10 times. And there's a special technique in performing a calibration. So if you want to learn more about this, I would recommend to listen to the webinar of my colleague, Dr. Ulrike Gast. She will have a calibration webinar on September 20th. So this is very interesting. You can go to eppendorf.com/etc to find out about our upcoming webinars. And additionally, she is giving seminars on calibration of pipettes where you can learn how to pipette correctly. And if you do not have all the requirements in your laboratory, I would recommend to send the pipette to calibration so that you're absolutely sure about everything has been done right and to get a new certificate for the pipettes.
Can dispensers be used for cell suspensions, or will cells settle out in them during a multi-dispense process?
No, sure, you can use a dispenser for cell culture and for cell seeding. We advertised the dispenser here during the whole webinar, and we already made some experiments on that with our repeater M4, which has been tested especially during cell culture experiments. And as long as you work in a steady manner, the cells will not settle down so fast.
And there's no difference between suspension cells and adherent cells. I think that was what the question was pointed out to.
How can you prevent ethanol from dripping out the tip when using air-cushioned pipettes?
Yes. When pipetting ethanol with an air-cushioned pipette, you will have to remember that, it tends to drip, that's right. So one recommendation I can give is to pre-wet the pipette tip. That means that you aspirate and dispense the ethanol 2 to 3 times. So just aspirate it and dispense it right back into the same vessel. So like this, you pre-wet the air inside the air-cushioned pipette and you reduce the dripping. Additionally, you can apply reverse pipetting. That's the technique I described a little earlier. And like this, you will avoid your air-cushioned pipette from dripping.
Which pipette system is more suitable for viscous liquids like glycerol?
Okay, so you mean which pipette system like compared to air-cushioned pipettes, I guess. So dispenser pipettes are working more precise when using viscous liquids. They do not have a delayed uptake of the liquid as you realize it with air-cushioned pipettes. And they are independent of influences like vapor pressure. So you will not have to consider special pipetting technique with a dispenser or a positive displacement pipette. This also exists. Therefore, I would recommend a positive displacement system for glycerol.
How can I make sure that the cell line I'm working with is not cross-contaminated with another cell line?
Well, you should authenticate the cell line. So this is done by basically taking a genetic fingerprint. So there's a method called STR analysis. STR stands for short tandem repeats. And especially when you work with human cell lines, there are databases available where you can compare the genetic fingerprint or the STR profile of your cells with the database, and then make sure that you don't have a mixed culture. And in general, you avoid the accidental mixing by simply work only with one cell line at a time in the biosafety cabinet, and also by having separate media bottles and tubes and aliquots for every cell type, and yeah, not mix it, of course, and of course also have separate tips, but this should be this you should do anyway in cell culture.
What is the maximum volume one can pipette using a dispenser?
Using a dispenser, the maximum volume would be 50 milliliter at once when using an electronic dispenser. So this is quite a large volume. And for an air cushion pipette, the maximum is only 10 milliliter.
Is there any alternative to heating media or thawing cells in a water bath, as this is a big source of contamination?
Well, for heating media or aliquots, you could use a thermoblock. This is a basically contamination-free system to pre-warm that. And for cells, it is a little bit different because cells need, as I pointed out, to be thawed as quickly as possible. And water warms up the liquid as quickly as possible. So, but also here it might be an alternative to use the thermoblock. I would just, again, test how sensitive the cells are to that longer period of thawing.
Looks like we have time for one last question. Is it mandatory to use the pipette and the tip from the same manufacturer?
Okay, that depends on what kind of laboratory you're working. So actually, if you're working in a regulated laboratory and you want to work after the ISO 8655, then it is mandatory, and it's ideal since the manufacturer of pipettes and pipette tips builds a complete system so that one works perfectly with the other. And only with the system provider who produces pipettes and pipette tips, you have the guarantee that even after autoclaving of pipette tips, the whole system still works accurately. So I would always recommend it.
I would like to thank our speakers, Dr. Jessica Wagener and Dr. Hanae Henke, for their presentations. Do you have any final comments?
Oh, just thank you very much for the nice moderation, and thank you very much for the audience for your attention.
Thank you also from my side, and only wanted to point out that we will get back to you if you send in a question. We will answer every question that came in during the webinar via email, so every question will be answered.
Well, thank you both.
Thank you.
I would also like to thank, oh, I'm sorry.
No, it's okay. Just go on, Judy, please.
Oh, no. Are you sure? Okay. I would also like to thank our sponsor, Eppendorf, for making today's educational webcast possible. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. Today's webcast will be available for on-demand viewing through November 30th, 2016. You'll receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Goodbye.
Bye. Bye.